session, I'll teach you how to draw a bacterial cell. As you all know, bacteria is a living organism and it is a prokaryote. That means it doesn't have a well-defined nucleus. It just have a region in the cytoplasm which has a circular double-stranded DNA, and that region is called as nucleoid. Bacteria doesn't have membrane-bound organelles, but it has cell membrane, cytoplasm, and a lot of enzymes. Let's start the diagram. Here we are going to draw a rod-shaped bacterium. So, let us first draw two lines. And connect them. Okay, so this is the basic structure of a rod-shaped bacteria. So let us assume that this is its cell membrane, because it's a living cell and it has a cell membrane. And inside the cell membrane, it has cytoplasm. So what you can do, you can use your pencil and smudge the area. So how do you smudge it? Just draw a few lines and smudge with your fingers like this if you're using a pencil. So smudge this entire area, shade it neatly. Okay. Now, at certain places, the cell membrane invaginates into the cytoplasm into loops like this, and this is called as mesosomes. They form loops like this, forming mesosomes. So this is a cell membrane, mesosome, and a cytoplasm. Now we have to draw the cell wall around this bacterial cell. This is the cell wall, and cell wall gives a shape to the bacteria and also helps in its protection. Okay. So this is the cell wall. Next is a capsule. So generally bacteria will be having a slimy coat around the cell wall, and that is called as a capsule. And capsulated bacteria are highly resistant and they are protected from the environmental factors, from extreme environmental conditions. So now here you can see three layers. So the inner one is cell membrane, this is a cell wall, and this is the capsule. Okay. Now let us draw the nucleoid region. Nucleoid, it doesn't have a nuclear membrane, but the chromosome is directly placed in the cytoplasm. And this DNA is double-stranded circular structure. So you can draw it like this, a double-stranded circular structure, which is the genetic material inside. Now along with this, it also in the cytoplasm, it will be having a lot of ribosomes. So just put some dark dots here which represent ribosomes. Now some bacteria will also be having flagella. So the flagella arises from here, and you can draw a flagella here. And what is the use of flagella? Flagella helps in movement. So this is a flagella of this bacteria. So sometimes only one flagella, sometimes a tuft of flagella will be there, which helps in movement. Now, on the capsule, you might also be able to see certain structures that are called as fimbriae. So fimbriae might be there in a bacteria. So few bacteria will be having certain fine finger-like or hair-like outgrowths like this throughout its outer surface, and they are called as fimbriae. The use of fimbriae is for attachment, especially during the time of reproduction bacteria. These are very fine, and fimbriae will be numerous number compared to that of flagella. Flagella will be either one or just few in tuft, whereas fimbriae will be numerous and the length is smaller compared to that of a flagella. Flagella will be longer. And as I said, the use of fimbriae is for attachment with another bacteria during the time of reproduction. Now, so other things which can be found, so these are the free ribosomes which are found. Sometimes even the ribosomes are attached together with RNAs and form a long strand like this. So this stretch of ribosomes and RNA complex is called as polysomes. Okay. So along with that, there will be different types of enzymes. For that, you can just put some dots here. So there are various enzymes that helps in respiration, nutrition, etc. Then sometimes some granules will be present, and storage granules especially. Storage granules can also be seen here in bacteria. So this is a bacterial cell. This is how it has to be labeled. This is the fimbriae, short fine processes which helps in attachment. This is capsule for protection. Cell wall gives shape and protection. Cell membrane allows permeability. This is mesosome, which is an invagination of the cell membrane. It helps in respiration. Cytoplasm. Here you can see the flagella. Polysome, it's a chain of ribosome with the RNA. These are ribosomes which are free in the cytoplasm. This is the circular DNA, which is the genetic material. And this region where this DNA is present is called as a nucleoid. These are the storage granule. And one more main important thing, most of the bacterial cells will be having a small extracellular DNA, which is called as plasmid. Thank you.